All right, so continuing with chapter 15, now we're going to go and look at the eukaryotic side of things. Okay, so eukaryotes. Now there's going to be a lot of differences between eukaryotic and prokaryotic gene regulation. In fact, remember, the genes in eukaryotes look kind of different from the genes in prokaryotes. We do not have operons in eukaryotes. We don't have operons. We don't have operators. Our genes are not clustered. Even if genes encode proteins that carry out similar functions or are imperative for the same metabolic pathway, the proteins are going to be, the genes that encode those proteins are going to be sparse throughout the entire genome. They're not going to be found in specific areas. For example, I don't know, our hemoglobin, for example, which is made out of the alpha subunit and the beta subunit. Alpha is going to probably be encoded in a gene on, I don't know, chromosome 7. I'm making this up, by the way. And beta is going to be encoded on a gene that's found on chromosome 15. Who knows? Again, I'm making it up. I don't remember which chromosomes encode the alpha and beta subunits of hemoglobin. But as you can see, this is just basically an example to show you that these two genes, their products are incredibly intertwined to make something that they are both needed to make one actual protein, and yet their genes are not going to be found next to each other. So we don't have clusters in the eukaryotes. We don't have operons in eukaryotes. Our genes look a little bit different. They still have a promoter. They still have a lot of actual added regulatory sequences that we'll get into later on. But they're definitely not clustered. Remember, our mRNA is monocistronic, not polycistronic. It's just one transcriptional unit coming from and deriving from one specific gene. So all cells in a multicellular eukaryote have a complete genome, even though all the cells have a complete genome, only part of that genome is going to be expressed. Remember, most eukaryotes are multicellular. A lot of eukaryotes are multicellular, so we have different cell types. And the way that we get these different cell types is by expressing different areas of the genome, right? So your liver cells, your hepatocytes, are only going to be expressing the liver-based genes and none of the other genes. Okay, so this is one thing. In addition, they have specific things that they only express at determinate amount of time, like at specific times. So, you know, insulin is only going to be made when you eat sugar or when there's sugar in your bloodstream. It's not going to be made any other time apart from that. So the growth and the development of eukaryotic cells is regulated to occur at appropriate places and at appropriate times during the development. If you lose this kind of regulation, this is what leads to cancer, right? This is what leads to cells becoming different kind, like a cell that has always been a liver cell is now all of a sudden forming features of another kind of type of cell. And then we have uncontrolled regulation of certain genes that are involved in allowing the cell cycle to go forward, and incredible inhibition of genes that usually stop the cell cycle to go forward. So we have lots of different defects and cancers that can arise from that. So regulation exploits in eukaryotes. Since eukaryotes are a lot more complex, the regulation here is going to be a lot more complex too. They exploit a wide range of ways to alter the expression of genes. Things like there's all sorts of things. So in bacteria, we mainly focus on transcriptional regulation. I did mention that it also has translational and post-translational regulation, but we didn't really focus on that much. But in eukaryotes, the regulation happens at multiple stages. In fact, it even happens in the DNA when it's just sitting in the nucleus as it is itself, right? Our DNA is associated with chromatin. We're going to see that there's different kinds of our DNA, when it's associated with proteins and therefore forming those nucleosomes and those really tight structures, is basically inaccessible to DNA replication machinery, to DNA transcription machinery. It's inaccessible, and therefore it's one way that we can regulate how and when our DNA can be transcribed or even replicated. Transcription can also be a method of or a place where we can have regulation, followed by, remember, in eukaryotes, we have our pre-mRNA that is basically packed full of intervening sequences called introns, and we have to remove those introns. In addition, we have to make actual modifications to the pre-mRNA, like add a poly A tail on the 3' end or add a 5' cap at the 5' end. So these places are another, like this RNA processing is another additional place where regulation can occur. So regulation of splicing and of processing. We also can regulate the actual transfer of the mRNA to the outside of the nucleus. So going to the cytoplasm. This could be another additional place where mRNA can be processed. In fact, a lot of times, because we have this added step, our mRNA compared to prokaryotic mRNA has a really long half-life, meaning it takes a lot longer for our mRNA to decay compared to bacterial mRNA. Bacterial mRNA can basically decay in a matter of minutes, in a matter of minutes, few minutes. And this basically kind of allows the bacteria to respond to its environment very, very quickly. While our mRNA, once we express our gene into and make an actual transcript, make an actual mRNA, it can last for a couple, even sometimes a couple of days. So a few hours. There's not that many times where it lasts a couple of days. There's one. But mainly it can last for a few hours. So a lot longer lifetime than the lifetime of bacterial mRNA. Then we have translational regulation as well, so we can regulate the whole process of translation. At this point, we've crossed a lot of different processes, so it doesn't happen as often because it's just not as efficient as, you know, inhibiting the gene expression right at the get-go or right at the beginning. Right now, we've gone through all sorts of things. We've expended energy going through transcription and moving the mRNA from the nucleus to the cytoplasm, but it's still a possibility depending on the different stimuli that we get. And of course, even at the very end, our own proteins can undergo modification, which can then further regulate them. So there's a lot of things to consider. We also have a lot more DNA compared to bacteria, right? And so because of that, obviously things get more complicated. Okay, so the first place that we're going to look at eukaryotic regulation is in terms of chromatin and chromosome organization. So there's two structural features of eukaryotic genes that distinguish them from prokaryotic genes. So the first thing is that, of course, our genetic material, our DNA, is found within the nucleus. It's found within this discrete compartment. And so transcription and translation cannot happen together, unlike prokaryotes. They can definitely not happen together. And so this is one of the main things that distinguishes eukaryotic genes from prokaryotic genes. And then the other thing that we have is that eukaryotic genes are always going to be wound around specific proteins called histones to form different bead-like structures called nucleosomes. And this wounding thing is imperative because otherwise all of the DNA that we have, the 3 billion nucleotides and the 3 billion base pairs that we have sequestered within the nucleus would not just only fit within a cell, but definitely not within a nucleus. So we need our DNA to be wrapped around all sorts of things, all sorts of proteins in order to let it fit. And so this association of DNA, so chromosome with protein, is known as chromatin. And the actual presence of the chromatin can be a physical obstruction, right? It could be, it can physically obstruct things from happening. So DNA replication, so the enzymes of like DNA polymerase, which needs to go in and actually find a free single-stranded DNA, cannot access the free single-stranded DNA because the DNA is looped around a specific histone. Same thing goes for transcription, right? So the structure of the chromatin is very inhibitory to many of these processes. So we need to make sure that we either rearrange the chromatin, right, in places that we want, like in places that we know need to be expressed, or even properly arrange the chromatin to make it even more tight and more packed so that those areas that have really tight and close chromatin association are not going to be read, they're not going to be expressed. So let's look at it. So the first thing that you actually might find interesting, I love this, because scientists used to think that the DNA when found in the nucleus was basically arranged as, was basically not even arranged, it was just a sloppy, messy bowl of spaghetti, essentially, or a sloppy, messy bowl of DNA in the nucleus, in the little bowl, in the nuclear bowl. But now they're realizing that it's actually not the case, that we actually start forming these discrete territories. The chromosomes start kind of looping, as you can see here. Chromosomes start looping and they form discrete territories in certain areas of the nucleus. And these are known as chromosome territories. So in the nucleus, each chromosome basically stays apart from other chromosomes. And in between the chromosomes, we have these interchromosomal domains that don't have any DNA, right? They don't have any DNA between them. And then they loop out and form these transcription factories. So these chromosomes that, or the areas of the chromosome that do go through transcription will loop out, that are necessary to go through transcription, that are always expressed, for example, that are constitutive maybe, will loop out and form these transcription factories. And we've noticed that in these particular areas, where they loop in. There's no other like messy DNA coming in the way. It's just that area, the chromatin, that is required to be expressed, that needs to be expressed. The gene needs to be made into the protein. And in this transcription factory, we have all sorts of transcription factors and all the RNA polymerases and all the goods that we need in order for transcription to occur. And it's just really interesting because they used to think that chromosomes were just sprawled, kind of like, again, a messy bowl of spaghetti. I wish I had some leftover spaghetti to show you guys. And so it was just very random, very messy, very all over the place. But in actuality, it's very organized. Chromosomes kind of stay to themselves, right? And then the areas that need to be transcribed, they're continuously looping them into these transcription factories, which happen not in just one, like not in just like the middle of the nucleus, if they happen in different areas around the nucleus. So this is like a nucleic feature where they contain the majority of the active RNA polymerase and other transcription factor. And they basically, they're little factories because this is where have steady, good, ongoing transcription occurs. So it's very interesting to note. So this regulation can happen here just in terms of organization of the chromosome. If the chromosome hadn't organized and hadn't looped out that specific region of itself into the transcription factory, it's probably very hard to actually transcribe that region. Another thing to consider is chromatin remodeling. So chromatin remodeling is an important step in gene regulation, and it basically involves changing either to the nucleosome or the DNA. So changing that whole association that you have with the chromosome and the protein. So you can either increase that remember, so the nucleosome are just 8 histones. So each histone has a bunch of DNA wound around it, and then you form 8 histones together to form one bigger bead. And then with those kind of aggregate even more together to form a 30 nanometer fiber, essentially. So very, very tight association with your DNA. So you can further close or tighten that associated DNA with the nucleosome, and this inhibits the DNA from the proteins, inhibits the DNA from being accessed by the proteins involved in whatever action like replication or transcription. And then you can further remodel the chromatin, basically allow the histones to kind of fall apart to kind of separate themselves from the DNA, to just loosen things up a little bit, move the nucleosomes around so that they used to be like covering this particular DNA right here, but now the nucleosome moves this way so that this DNA is now free and available and accessible to other proteins. And so now we call it that like opened, right? So in order for the transcription to happen, the chromatin needs to be opened. Couple of words that we use to describe this chromatin. I'm not sure if it doesn't come up here, but I'm going to say it because it's important. Heterochromatin is essentially when your chromatin is really heavily packed and condensed, right? And so your DNA is in a very tight, close structure. And usually when your chromosome is in that area that has heterochromatin packing or association, there's no, basically transcription doesn't occur. There's no way you could read your genes. It's only if there's remodeling and we go from heterochromatin to euchromatin. Euchromatin is your DNA is a lot more open, and so you can actually have transcription and replication occur, right? So areas that do not want to be read are going to be usually in heterochromatin format, while areas that are actively read and are actively transcribed are going to be in euchromatin format. So an example would be the X bar body in females, right? The X chromosome that we know is one of the X chromosomes is always going to be shut down in each cell. One of the things that it does is that it forms that heterochromatin tight structure so that the DNA literally becomes inaccessible to the proteins that are necessary for transcription and DNA replication. We can also modify the histones. So remember, the histones are the proteins that the DNA wraps around in order to form those nucleosomal kind of beads. We can basically remodel them by modifying the histones. So the nucleosome modification can occur in 3 ways. We can change the nucleosome composition. There's different kinds of histones. So depending on the histone itself, each histone has different affinity towards the DNA. So with variant histones like H2A.Z, this affects the nucleosome mobility and positioning of the DNA, thereby affecting the transcription. So H2A.Z is a little bit more, has less affinity than a histone like H2A, for example. And so that particular histone will make the DNA a little bit more accessible. We also have histone modification. This is when we are directly changing the histone. So we add specific chemical moieties like acetyl or methylate. We can methylate our histones as well. So we can acetylate or methylate our histones. And when we do that, we actually change the chemical nature of the histone, therefore making it either making it have more affinity towards the DNA or less affinity towards the DNA. So for example, acetylation actually makes a histone have less affinity towards the DNA, because acetylation is making that histone, which is normally positive. Histones have lots of histidine in them, lots of that particular amino acid, and they're very positive, which makes sense because our DNA is negative. So our negative DNA is going to wind around these positive little beads or these positive little proteins. But the acetylation of this histone by the enzyme histone acetyltransferase enzymes, or HATs, these enzymes, basically, when they acetylate the histones, they make it less positive, right? And so by making it less positive, they have less of an attraction to the DNA, less of that strong affinity that they normally have. And the DNA starts kind of, leaning out of the, leaning away from the histones, as you can see here. The part of the histone that gets acetylated or methylated or whatever are usually the histone tails, which are always found protruding outside of this little DNA nucleosome that we have formed. Okay, so we can make the DNA a little bit more accessible by acetylating our histones, making them a little bit more negative, less positive, so a little bit more negative. And this way our DNA kind of does not like that. We can also do the opposite, right? We have histone deacetylase, which basically gets rid of the acetyl chemical group from the histones. And so by getting rid of the acetyl chemical group, we make the histones their proper positive cells, and therefore the DNA is going to really safely wrap around them like nice and tightly. They can also be modified by phosphorylation and methylation. Usually methylation of histones basically makes the whole chromatin association even tighter. Methylation is usually something that we use to inhibit transcription or inhibit DNA replication. It's it has a very negative effect. So by unmethylating, we are allowing transcription or allowing DNA replication. But there are some exceptions, as there is always in biology, right? And then we also have chromatin remodeling complexes. So these are basically very energy dependent pathways or very energy dependent procedures that literally reposition or remove nucleosomes in the DNA. So a DNA could be wound around in this specific arrangement where we have nucleosomes right next to each other. Note how there are 8 histones per each nucleosome bead. We can have a chromatin remodeler that can come around. So these are just large multi-subunit complexes that use a lot of energy to arrange nucleosomes along the DNA. And so they can then arrange them and kind of expose different parts of the DNA. So now they slide away from each other, and this sliding exposes the DNA, right? You can pull off the DNA off the nucleosome, or these nucleosomes can actually split and they form nucleosome dimers, and then all sorts of different loops of DNA can now become available to whatever machinery they need to, whatever process they need to actually go through. So the nucleosome remodeling complexes may alter the nucleosome structures in several ways, including altering the contacts between DNA and the histones, alternating the path of the DNA around the nucleosome, and altering the structure of the nucleosome core itself, like we did here when the core particle just basically dimerized. So we got 4 to 4, right? So all of this is chromatin remodelers and very energy, it's a very energy dependent process, and you're just loosening the attachment of the DNA to the histones or vice versa, because as you can see in all of these chemical reactions, the arrow goes both ways. And then finally, we can also directly affect our DNA, right? We can actually directly change and tweak the DNA base pairs. A lot of the times we use cytosine becomes basically as a culprit is something, the base that we usually methylate a lot. So DNA methylation is adding the methyl group CH3 directly to the DNA. A lot of times, again, it's cytosine. It's the addition of this methyl group. And this basically shuts things down. We've actually seen this already. We saw that methylation could happen it could be heritable. We have different methylation patterns that happen in the mom or in the dad, and they could be inherited into the offspring. And even though the gene was perfectly fine, the fact that we had methylation to shut the gene off. So this is, again, underlying how important regulation is. The fact that we had methylation in the mom that shut the gene down, and that methylation pattern was carried on and inherited into the offspring, now that offspring cannot express that gene either. So there's an inverse relation between the degree of methylation and the degree of expression. So the more the methylation, the less expression. The less methylation we have, the more expression we have. The more expression from gene to protein, so transcription and translation. So the methylation patterns are tissue specific, so different tissues can have different amounts of methylation. Usually it's a little bit less than 2%, all the way up to 7% of the genome in the tissue being methylated, which is a substantial amount of our genome. Once this is established, again, once the methylation pattern is established, it can be inherited from cell to cell. So even our own cells, when they undergo mitosis and they're just duplicating themselves within the same organism, the methylation pattern that the original cell had can be passed on. We see this also, again, I'm bringing this up again, but with the X-bar body, right? So the X-bar body, another thing that they have is that it is methylated. Another thing that really shuts down the extra X chromosome, not the extra X chromosome, because that insinuates that or implies that we have too many. But the second X chromosome that women have is basically another way that is shut down on top of the heterochromatin, on top of, if you remember, it actually has RNA that kind of like goes against it and shuts it down, basically binds to the DNA, which so making it further more inaccessible. But it also has, in addition to all that, methylation of some of its DNA bases. And this is one of the ways in which, like, further, like, cells that originate from that original cell that has the, X chromosome number 2 has become the inactive bar body. All the cells that come and like are progenitors of that initial cell will have X chromosome number 2 as their inactive one, and not by chance X chromosome number 1, right? So there's proper methylation patterns, and these are essential for development, and they're essentially like inherited, not just in the body through mitosis, but can also be inherited through meiosis and obviously egg and sperm fertilization and normal offspring progeny. So it's repressed transcription by inhibiting the binding of transcription factors. So how does methylation, how does it do such crazy like inhibitory work, right? The reason why it does so is because it's basically in the way. It's bound to these bases, and the bases are no longer accessible to the various transcription factors and other proteins that are necessary for DNA replication or DNA transcription. It can also accrue repressive chromatin remodeling complexes. So what we saw before, so chromatin remodelers are basically repressed and go backwards, so they form more tighter associations of the chromatin. So yeah, so this is basically essentially how DNA methylation works. So we looked at the organization of the chromosome and how that works in regulation of eukaryotes. And then the next section we'll look at is the actual genes and the cis-acting sites, so the regulatory sites found within the DNA, within the genome, that act in a regulatory way as well. So I'll see you guys in the next video.